Very good morning, everyone. On behalf of AARI Chennai, I take this immense pleasure of welcoming you all to this bio webinar session 3. Today we have our special guest, Dr. Vaibhav A. Mantri, Head Applied Phycology and Biotechnology Division, Central Salt and Marine Chemicals Research Institute, Bhavnagar, Gujarat. Sir is one of the well-renowned scientists in our country in the field of applied phycology. Before moving to Bhavnagar as a principal scientist and head, he was working as a senior scientist at Marine Algae Research Station, CSMCRI Mandapam Camp. He successfully executed several nationally important, highly funded projects with various funding agencies like Ministry of Environment and Forest, CSIR, DBT, DSIR, NFDB, and also a project funded by Tamil Nadu Government Fisheries Department. Sir acted as a key person for bringing several international collaborative projects to CSIR. He is also an editorial board member of International Journal of Geology, Earth and Environmental Sciences. He has more than 2 decades of experience in algal research. He acted as an examiner and also guided several PhD candidates. Several research fellows and MSc students were also got trained with Sir's able guidance. Needless to mention of this renowned scientist's social responsibility, upon recognizing this, CSIR awarded him the Science and Technology Innovation for Rural Development for Seaweed Cultivation and Value Addition in 2013. He is also a CSIR Raman Research Fellow. And for his academic excellence, he was awarded with several National Merit Fellowships. He has numerous peer publications in national and international journals. He also has US patent for innovating cost-effective, efficient device for large-scale seaweed harvesting. And several national patents are awaiting for the same. He also co-invented cost-effective process of agarose preparation from Gracilaria species, and international patents are awaiting. The achievements of him are numerous to enumerate with this short time. Without further delaying, I take the honor of welcoming Dr. Vaibhav A. Mantri for delivering his speech on seaweed diversity of India. Sir, we are eager and excited to hear from you.
Hello. Thank you very much and good morning to all of you. Where I am audible?
Yes, sir. You are audible, sir.
Hello.
Yes, sir. We can hear you. Please.
So, I am very much thankful to AARI for inviting me and giving me the opportunity to speak before all of you. And I have decided to talk today on very interesting topic, and to me it is why it is important because biodiversity or taxonomy is the field which is basically very neglected, and I'm sure many of you will agree with me that in India doing biodiversity studies is very difficult because of funding constraint mostly, and there are a lot of challenges and still there are a lot of opportunities, and all these I would like to touch on today. And therefore before starting this seminar, let me first thank Director CSMCRI for allowing me to speak on this occasion. And please note that whatever I am talking today is not my work. It's a work of several scientists and students, and not only of only CSMCRI, but as a whole country. Those all who are working on biodiversity, I tried to take some of their good things. And with this brief background, let me start my lecture. Many of you might be aware of CSMCRI. But still, let me say a few words. It's a CSIR laboratory located at Bhavnagar, Gujarat, and you all know that CSIR has several laboratories and their mandate is different. And this particular laboratory basically was established after partition, because most of the salt producing areas went to Pakistan, and country was facing lot of trouble for producing salt itself. And you all know that salt is very important and essential commodity. So that's how CSMCRI was born. And although I'm going to talk about seaweed, it comes under chemical sciences cluster. And as I said, the main mandate of the institute when it established was researching on salt. And slowly as our organization grew, we started working on various aspects of sea. And seaweed being one of the plant which grows in seaweed, that also came into main mandate. And today CSMCRI works on lot of other fields like membrane science, desalination, and wasteland reclamation, etc. As you all know that seaweeds, when you listen, it has very negative connotation, like sea and weed. So basically I would call them as marine algae, not seaweed. But I have used term seaweed because it's a very widely used term. But please note that they are nowhere related to weeds. And if you see, they are basically categorized. I would not say classification, because the term classification these days has no meaning. Everything is phylogeny. Everything is seen in terms of evolution. So this important marine renewable resource is categorized artificially into three groups, mainly green algae, brown algae, and red algae. They call it as a chlorophyta, phaeophyta, and rhodophyta. I have shown you some of the pictures, and as I said, it's marine algae. I have chosen first picture, left hand side, extreme left hand side. You will see it's not a seaweed, but it's a chlorella species. And chlorella mostly is a freshwater algae, but also some marine and estuarine forms are also present. So apart from chlorella, all are basically macroalgae, marine macroalgae. You can see that most of the green seaweeds, many of you in colleges or during your curriculum might have seen at least few of them, like alva and sargassum and gracilaria. These are mostly very commonly shown during our curriculum, but it has so much diversity. You can see, I'll be shortly covering our Indian scenario, where we have close to 1,000 different seaweeds. But just to give you a glimpse how they look, I thought of sharing this slide. Okay, so when you see the historic account, basically seaweeds are have attracted attention of world scientists basically in early and mid 1800, means you see how many years back. And it all has started with natural history, basically. And natural history, people started looking at various forms, flora, fauna, as well as marine forms. They started studying seaweeds. And I have taken some of the old pictures. You can see one of them below is from Natural History Museum, and how people is to study. You can see they is to collect and make drawings. But if you see the world scenario, especially after First World War, and to be very specific, after Second World War, people trying to see look at seaweed for industrial point of view, because lot of destruction has happened. And people were trying to see alternative resources for many of their requirement, including soda ash, glass making, and agar, and so on and so forth. And also in India, after World War II, there was a lot of dearth of getting basically agar. And agar in those days is to be used for making cholera vaccine. And active steps were taken, and Board of Scientific and Industrial Research, basically, which has given our country CSIR, and it is started looking at seaweed very seriously. And the research department at University of Travancore, people worked on gracilaria. And you all know, many of you must be knowing Mrs. Francisca Thivi. She is, I would say, a lady who basically popularized seaweed research in India, and she worked in US, and she started developing protocol for agar extraction. Now, with all this historical background, let me come to the today's global seaweed industry. Many of you will be very much surprised to know that uh, world today produces almost 31 million wet tons of seaweeds, which has a value of 12 billion US dollar. And you will be very much again surprised to know that most of it comes from 5 to 6 genera, not more than 7 genera, I would say. You can see here that the most important contribution is made by Euchema and Capaficus. These are basically red seaweeds, Euchema, Capaficus and Gracilaria. They form more than half of the percentage of production. After that, it comes basically Laminaria and Undaria, and they are basically a brown seaweeds. And also, if you see, these are primarily the phycocolloid yielding seaweeds, and if you see, they are basically make volume. But when you see about value, most of it comes from porphyra. Many of you might have heard the name nori, and there are a lot of sushi restaurants coming in big cities, and porphyra makes basically a big contribution towards the economy. When you see, now let us go to India, how it has started, and I'll be showing in next slide, which was the first seaweed described from India. But before that, you all have to see that there are basically various stages. As I said, the naturalists which basically came to India, either they were European missionaries or British officers, they all had taken a great deal of interest in collecting various forms of seaweeds. And those days they used to collect, and all were not basically experts in seaweed, but they used to make herbarium or drawings and pictures, not pictures actually, but drawings, and used to send them to various experts throughout the world. And that's how knowledge about Indian seaweed grew. And then some of them have trained Indian people as well, and some Indian people like our legendary Professor MOP Iyengar, he took great interest, and he started collecting and describing. So in third stage, basically our own phycologist of the country started studying seaweeds. Also, lot of expeditions, as many of you might have known that there were expeditions, and those expeditions I will not read, you all can see in the slide, but some of them like Sigboga was very very important. And all these expeditions basically started collecting seaweeds from seas around India, and they basically house lot of our collection in British Natural History Museum. Now, as I said, you can see that Indian phycologists who were very important to the country. As I said, Professor Iyengar was the first algologist who studied
Excuse me, sir.
Crucidae Island flora. Hello?
Excuse me, sir. Yeah, it's Perumal.
Yeah?
Sir, can you make the slide full screen, sir? Because this
Um,
partition, like you can.
I don't know how to do it.
You switch on the auto rotation and keep the mobile vertical. In your mobile, switch on the auto rotation.
I think let me speak, and I will share these slides with
Okay, sir,
people.
no problem. Then it's good, sir.
Okay?
Sir. Thanks,
Yeah.
sir. Yeah.
Yeah. Thank you.
Thank you, sir. Continue, sir.
So, Professor MOP Iyengar basically studied Indian seaweed flora, and those who are from South India or Those who are not even from South India, I'm very sure they might have heard the name Crusida Island. It's a very famous island, very beautiful island. When you go from Pamban to Rameshwaram, you can see it on right hand side. And this Crusida Island harbors a great seaweed flora, and he has studied all this basically in early, I would say, 30s. And then, Borgeson. Uh, Dr. Borgeson was basically a Danish prince and also basically a naturalist. And he was a friend of Professor Iyengar and visited India on the invitation of Government of India. He visited almost most of the places of seaweed rich places of our country, and he studied and published a series of papers in various journals, and he has described a great deal of Indian flora. As I mentioned, Mrs. Francisca Thivi, she was also a great taxonomist of the country and described several new genera and new species. Besides that, Professor Krishnamurti, I'm sure many of you, those who work on seaweed, might have met him, and he basically worked and published several volumes, and I would say maximum contribution was from Professor Krishnamurti as far as seaweed taxonomy is concerned, and also Professor Uma Maheshwar Rao, who was from Andhra University, who was basically initially worked on CMFRI, and he also has contributed as latest as his book has come in 2009. So I have not taken name of many algologists, but I just wanted to give you some idea about who were the pioneering people in the field. This particular picture, I just wanted to tell you that people were studying, but a lot of specific efforts were taken when Seaweed Research and Utilization Association of India was born. And this the first meeting which happened at Marine Algal Research Station, our own field station at Mandapam, on September 6th, 1970. And in this meeting, you can see Mrs. Thivi is sitting, Professor Krishnamurti is sitting, and I was told that even a person, cellulose company, which was in Ahmedabad, all the way went to Mandapam to take part in this meeting, and they decided that we should study seaweeds, holistic, not only taxonomy but application of it, and that's how our research basically started. Now let us see that which are the centers, very important for Indian seaweed research, and especially taxonomy. You can see that there are not very many centers, but certainly I must take name of Delhi University, Andhra University, and Pune University, from where I belong, CSMCRI of course, and Kerala there is a group of Professor Panikar in SN College, Calicut University, then in Orissa also there is a group in Behrampur University, and Tamil Nadu already you all know there is a CMFRI, Madras University, Calcutta University also worked on Professor Ruma Pal's group work on seaweed, and of course NIO in old days people used to work on seaweed. So with such a big country having several coastal states and union territories, there should be many more centers which should come, and these were basically the centers where a lot of studies have taken place. These are some of the literature I thought of sharing with all of you, those who might have not aware that CSMCRI in recent days has produced lot of books, and which are of very important to application and taxonomy and cultivation. As you all know, the first checklist of India was published from CSMCRI, and in 2001 we have published a revised checklist, and there is a book called Seaweeds of India, which was published by Springer, and that was also published. Then there are two volumes on Indian marine algae. They are basically given the state of the art research in several fields of recent advancements in applied aspects, published in 2006. And also there are a lot of manuals for cultivation, and you can see also some Hindi, recently some books in Hindi, brochures and small booklets were published, so that all this information should go to common people. When you compare Indian seaweed biodiversity, you would see that there are five major floristic, not actually floristic, a checklist kind of literature available. And you can see I am not going to read everything, and you see that I have also included yellow green and blue green algae. To be very honest, they should not be considered as a seaweed, but some of the authors have included them, so I thought of including it in this table so that you will get a clear picture. So as of now, a recent book published by Dr. P.S.N. Rao and his associate from BSI enumerated 1,100 1,019 species of marine algae. Okay, and composition and species wise and genus wise composition is given. When you see all this, you will definitely see that red seaweeds form a big proportion of the diversity. We'll come shortly why it is. Let us go to comparison of seaweeds of India versus world. See, these figures I have taken today morning only from Algabase. Many of you might be knowing that there is a Algabase site which is a database, and you can see that we have very meager. We represent almost 5% of world's seaweed diversity. When I saw a Ministry of Environment and Forest website, I got information that when you see the higher plants, basically we represent only 2 to 4% of land area of the world, but our diversity is 7 to 8%. But when you see ocean is a big part, but still we have only 4.7% of world's diversity. So it is very important to think why it is. So one of the reason is, of course, we don't have much research going on biodiversity, and we all, especially the young researchers who are listening to me today, should decide to take up this as their PhD topic or their research topic, and that's how we can basically explore our diversity. Now, you all have to also understand that although I showed five checklists in my previous University was the first who basically compiled this information, and he has published that paper as early as 1968 in uh, University Journal of Bombay University Journal, and he has compiled list of 411 different species from India. Now let us see what are the gaps and challenges in biodiversity studies. I am sure that whatever you have seen my previous slides, they are all historical, and what happened earlier and previous people. So all this is old, and now there is a need to do precise assessment of current biodiversity. What is happening today? I just now told you about Dr. Dikshit. and you see how old data is there. And also I said revised checklist of CSMCRI also 2001, and BSI checklist also 2012, and we are almost now have a decade old information, and we must study what is current biodiversity, and not at regional level or one small area. We have to have it at national level. We also do not have national database. As I said, Gary and Gary, that alga based database, you can see it's a world database is available, but we don't have a clear and exhaustive national database. There is a diminishing expertise. As many of you know, we don't have many taxonomists, active taxonomists. So the taxonomic expertise is fast depleting, and we have not done survey of several unexplored area. If you see the Indian context, many of the research is centered on Crusader Island, Kanyakumari, Okha Dwarka, or name few locations only. But people have never gone beyond these areas and studied new areas. Also, we don't have much research on recent approaches which are molecular based taxonomy. We have very few, and there is a group active group at Central University of Punjab at Batinda. Felix Bast is working with his student, but I don't see not many people are working. So we don't have also a focus on conservation. We basically, I'm going to come on shortly about endemic seaweeds, but we don't know basically what is the threat perception to those endemic seaweed, their IUCN status, nothing is known. So that is something is very big gap. Also, we don't have networking, and people are working in isolation. There is not a single program in India which is a networking program on seaweed. And we also have to conceptualize a big program, very big program. So these are some of the gaps I feel, and there may be many. People can basically debate. See this slide, I just wanted to show you very interesting analysis which we did a couple of months back. uh, we decided to see how new species are described from India. We took a literature and we saw decade wise how many new species were published, and this is basically up to last year 2019, and you can see that very dismal performance, very dismal performance. And you can see in a decade between 1930 and 40, and also 80 and 90, there was two peaks. And basically these two peaks were because, as I said, Borgeson came to India and studied seaweeds and described in series of publication, and that was the reason why between 30 and 40, that decade, new species were more. And also there was a funded project by Department of Science and Technology to Professor Desikachari, and Professor Desikachari with his two colleagues, eminent phycologists, Professor um, uh, Krishnamurti Sahab and uh, these people have worked basically and published two volumes, and these two volumes of Rhodophyta book also again showed a peak, and that is why these two peaks you can see. And also, as I was telling, I saw an old literature, and Amphiroa was the first seaweed described from India. I have given a photo, and you can see it was described in 1672. Now with this slide, I am sure you will see that we are not adding new species to the Indian seaweed flora. As I said, let us go one by one, gap areas which I mentioned. So CSMCRI has taken an initiative under 12th 5-year plan project of CSIR, and chose two basically states. One is Gujarat and another one is Tamil Nadu. and I am giving Gujarat data because already book is published, and my colleagues at Marine Algal Research Station Mandapam are shortly going to uh, you know bring a new book. It is by Oxford University Press, and that is about Tamil Nadu flora. But let us see what shows Gujarat shows. So under current seaweed biodiversity assessment, you can see several of locations of Gujarat were studied for 3 years, and there are almost 200 species my colleagues have reported, and you can see that these 198 species belong to uh, 62 from Rhodophyta, 54 species, and 23 genera of Chlorophyta, and 16 genera of Pteophyta. So basically, why I showed this slide here is, you can see when you see the current biodiversity assessment, you will see very different picture than whatever secondary information is available. These slides are just to show how these species are described in the book. Okay, so I showed green, brown, and red. So all lot of information is given. The taxonomic name, latitude, longitude is given. You know what kind of substratum it prefers, occurrence, abundance, its description. You know, and wherever uses and wherever available, gene sequence database also is given for these species. So one has to study basically various new locations, and current biodiversity assessment one has to do. Now this slide, as I said, there are 125 endemic species are identified from our country. That means these species basically have restricted distribution. They are not everywhere. You cannot find them basically outside India. And that's what I was telling in the beginning. Although we know which are endemic, we don't know what is the threat, whether their population still exists, and if it exists, in what area. So one can consider all this into giving various categories for conservation. That is not there. Now, as I said, national database, I have given here a couple of examples of global databases. You can see that there is a census of marine life, a global association of researchers, where basically there is a database available for marine, including seaweed. Ocean Biogeographic Information System, that is also again a global database for marine species, including seaweed. There is an exclusive database for seaweed called AlgaBase, and is run by uh, uh, Professor Michael Gary and his wife. And it is such an important resource that one can simply go to this database and find out what is the taxonomic status of particular species. Synonyms and references and photographs, everything is available. But such global database, exhaustive database is available. I just thought of telling, this is morning figure, that it has more than 1.5 lakh species already included, including infra-specific names, and it has more than 22,000 images and 61,000 bibliographic atom, and all this is 4 lakh distribution record. See how exhaustive, and this couple is maintaining this. But for a country which has so much of baseline data, we don't have a good database available. But I would not say that there is no database, because National Institute of Oceanography has developed some database and it is available. Also, National Chemical Laboratory has developed a software and a database called Sampada. And also, database India Algae was very recently created by Central University of Punjab. But my problem here is, although we have these 2-3 databases, they are not regularly updated. There is no new information. Probably they were a part of project. That is why we should have, as a nation, a single database where people should keep on adding information, and our country certainly needs such kind of database. As I
Sir,
said, diminishing expertise.
sir,
Hello? Sure. So basically, as I was telling that we also have to learn that how these people in the world are, working for capacity building. And as I said, this was a very famous program on basically international workshop on taxonomy of economically important seaweeds, which was supported by California Sea Grant College and handled by Professor Isabella Abbott. And also there is another course which is still going on, and I'm sure some of you might have also know it and might have participated, is called Marine Botany Summer Course at Friday Harbor Laboratories of University of Washington. With these two programs, India also should take some inspiration and do some capacity building training programs. I am very happy to say that CSMCRI has undertaken some of such programs at our center at Mandapam as well as at Bhavnagar. And the first program was in association with Krishnamurti Institute of Algology. You can see here Professor Krishnamurti in the photograph, and he was kind enough to come all the way to Mandapam and teach people, basic techniques of seaweed identification. And also CSMCRI Bhavnagar is conducting now under CSIR capacity building and training program courses on seaweed farming basically and value addition. And also I'm very happy that Botanical Survey of India, Dr. Palaniswamy has very recently under ICOPTEX project has conducted one such program at Coimbatore. But what I wanted to say here is that these are good beginnings, but we also have to pursue with funding agencies and with industry rather, because all this expertise is needed ultimately for whom? Industry. My last slides are going to come to show you how this taxonomy is helping industry. So basically, with all these efforts, we must do some capacity building, programs in the country. Now, this slide I have purposefully taken. This is my personal work, and under one of the 12th five-year program, we have basically studied one island called Kalugar Island in Gulf of Kutch. You know that in Gulf of Kutch and in Gulf of Mannar there are islands, mostly coral islands, and these harbor a lot of seaweed diversity. And many of the islands in Gulf of Mannar were extensively studied, but not many islands on west coast, especially Gulf of Kutch, were studied. And we thought of studying these islands under a program funded by CSIR. And this Kalugar island was studied during 2009 and 10. And you will see that very interestingly that in only couple of visits we could identify two very great discoveries. One is a record of male gametophytic plant in Dictyota serrata. That was first time in the world. So as many of you might be aware that seaweeds has a life cycle, typical life cycle, and they have male, female gametophytes and tetrasporophytes, and either the gametophytes or sporophytes are identical, or all are identical, or different. So basically these phases are different, and male gametophytic plants in Dictyoteris serrata were not known, and we have identified them from these studies. And also and there was a new generic record, and we have made, and that is again from family Sargassy, and it is yet to be published, this particular thing. But what I wanted to say that you can see that with such a few visits we could identify a lot of good diversity. And you can see here that this mapping of seaweed diversity has resulted into 93 new seaweed records, and of which uh, 11 were new to India and 14 were new to Gujarat. So I think, as I said, we must go to new locations to really find out the diversity. Now, one of another gap areas which I said was basically of molecular studies, and I'm going to give you two examples. As I said, Dr. Felix Bast is working with his students, I know, but these two examples I am going to give basically where a monographic kind of work is done. Like in case of one of our past student, she has studied a Gracilaria, one of the diverse seaweed genus in red seaweed, and she has visited various places and collected lot of Gracilaria species from India, and she has used basically three markers, molecular markers, 18S, then ITS and Rubisco, so and Cox spacer. So basically she has used nuclear marker, a mitochondrial marker and chloroplast marker. So with this she had shown the phylogeny of Indian Gracilaria species. Another student from our institute has also worked on Caulerpa. This is another important and diverse genus of Indian seaweeds, and he also has used various markers, 4 different markers. I think here tough A gene also was included, which was not included in Gracilaria, and he found out that this tough A is also a better marker to study, and very interesting observations have come from this study. As many of you might be knowing that there is a Caulerpa veravallensis, again is a I will not say it endemic because now it was reported from Pakistan and other areas, but its origin was from India, and he after studying this he could he could conclude that there is a sister relationship between Caulerpa veravallensis and Caulerpa racemosa variety cylindrica. Also, he told that after this research, he concluded that Caulerpa microphysa and Caulerpa lentilifera are not two different species but a single species, a one species. So you see how interesting this is, and one has to use these new techniques. Now when you see molecular studies and diversity, one should not restrict them only up to species level. This particular slide I am showing, as I said just now, that algae has various life cycle stages, and studying differences between life cycle stages also a part of biodiversity study, and it is useful for bioprospection. I will be coming very shortly. This example is again given in one of the industrially important species called Gracilaria dura. This species, basically why it is important, because it directly gives agaros. Again, it has a restricted distribution only at wherever at very near to Somnath. Some of you people might have heard Somnath. It is very famous pilgrimage place, and this particular species is only available there. And apart from this, there was only one record of Professor Uma Maheshwar Rao of this species from Kanyakumari. But except Professor Uma Maheshwar Rao, nobody has collected it. But from wherever, certainly you can collect. But the whole collection, I don't think can go more than 100 kilo. But it's very important because it gives directly agaros. And for those people who doesn't know how agaros is prepared, just for your information, basically big industries, they get agar and low gel strength agar, and then they process it. Basically they remove sulfate content from that, sulfate moiety they remove, and then it becomes a high gel strength agaros. It's very costly technology, but our institute, my colleagues have developed, and we got a US patent also, that this particular seaweed, by treating the procedure which we have developed, can give directly agaros. So there is no need of purification, and that's how this particular species is very important for commerce. Now a biodiversity, as I was telling, should not be looked at only taxonomy and all. You can see here we have male gametophyte, female gametophyte and tetrasporophyte, and it shows a various different kind of agarose characteristics. Basically yield, gel strength, viscosity, they are very important. We have also worked on its cultivation, and we have published couple of papers, and we have one manuscript which is ready to go tomorrow, and where we have shown how one can select a specific life cycle stage basically for farming, because this technology has been taken by Messrs. Aqua Agri from our institute. This technology is already transferred, and we have also developed ISSR markers for identifying good uh, trait, good traits from this, especially tetrasporophyte, which gives a good basically yield, and gel strength as well, and it also grows faster. So what I wanted to say in this slide is biodiversity can be even under important for studying life cycle stages, within a species differences. So that's how these life cycle studies are different important. Again, as I said, we this is also not published, but just for your information, that we have got one new codium species to whom we have named spiralis. You can see it, spiral arrangement, and this kind of information is very essential when you basically, and as I said, this was again from that island. So this, when you go to new areas, you can always have a chance to find new species. Now, why these taxonomic studies are important? As I said, this is another example. One of the endemic species, called Alva ovata, or it was described as Enteromorpha ovata, but we have worked, we are yet to publish, we have transferred it into Alva. Many of you might be knowing that now there is no more Enteromorpha genus. So this basically species grows at a place called Gopinath, very near to a largest ship breaking yard in the world called Alang. Those who are from Gujarat might be knowing that Alang is the largest ship breaking yard, and it breaks the ships. And this particular species is very near to, within a 50 kilometers radius of this place. And so this, we have to have conservation for this. Now what we discovered in this, I have given a life cycle, typical life cycle. So typical life cycle in Alva has male and female gametes. They come, they fuse, forms zygote, and then there is a sporophytic plant which again goes and produces zoospores, and the zoospores germinate and form again male and female gametophyte, separate plant. But now is a elaborate, you need to have male gametophyte, you need to have female gametophyte, all these you need to have to conserve it in laboratory. But one of our studies shows that it also has a asexual life cycle. So there is no need to go to male gametophyte, female gametophyte. Asexually, sporophytic plant can grow. That is our discovery. So it is very important discovery, because you can basically encircle this species in the laboratory only through asexual propagation. So why I gave this example, that when you study a biodiversity, you must you must basically study it in a holistic manner, and life cycle studies also is a part of biodiversity and can be helpful for conserving these species. Now, second important thing for conservation, I think, is another area called tissue culture or micropropagation. This is one of the works of my senior colleague, Dr. C.R.K. Reddy, who is a well-known phycologist for tissue culture work, not only in India but for world. You can see that we were successful in developing basically a callus and also a somatic embryos in some of the species. And how it is again useful beside conservation is, this tissue culture progeny was cultivated in the field, and I have shown here an example of Gillidiella acirosa. Many of you might be aware that Gillidiella acirosa is the best source of bacteriological grade agar in our country, and there are 23 agar producing countries which are exclusively depend on this particular species, and most of our biomass comes from basically natural collection of Gulf of Mannar Islands. And my colleagues at Mandapam, Dr. Ganeshan, has developed a technology for its cultivation, which is transferred to a leading industry, Snap Alginate. And so these kind of basic research can help, you know, to industries as well. As I was telling you, that this biodiversity studies and basic research is very important. Now, this is again very important. We have under herbarium center in the country, and you can see that basically we have more than 250 different species, and we have more than 5,000 herbarium specimens now, today, as of now. And now it is located, it was established at Bhavnagar, but our management has taken some decision, and now it is at our center Mandapam. And our scientists, basically, you know, when some of you want to write a paper, many of the reviewers ask whether you have deposited your any kind of work you do, they ask whether you can basically deposited your collection. So our scientists, our staff, we give this kind of opportunity. You can basically deposit your collection in our herbarium center, and we give you a accession number which you can basically use it for your publications. So this is what we have developed, and it is at Mandapam, and one can contact scientist in charge, Marine Algae Research Center, Dr. Ishwaran, for this. I think I am at very end of my lecture, and just would like to show you that all this taxonomy ultimately has a big opportunity in farming and processing. And I have just shown you a couple of photos where you can see that many of you might be aware that commercial cultivation is happening in India today, and we have all these well proven technologies, viable technologies. Some of them have even transferred to, as I said, we have transferred Gilidiella technology to Snap Alginate. We have transferred Gracilaria dura technology now to AquaAgri, and many of the leading industries are now in touch with us for basically agar and edible seaweed, especially Enteromorpha and Alva. So all these biodiversity studies are ultimately helping our country basically to become a self-reliant. You know, you will be very surprised. I have not included that information because my focus was on basically on diversity and taxonomy. You know, we have three polysaccharides: agar, alginate, and carrageenan. You know, the agar requirement is something around 600 metric ton per annum, and we only produce 300, 200 something metric tons. Imagine our requirement, not even 50% requirement is met. When you see alginate, our requirement is something around 1000 metric tons per annum. Here also again we don't produce much. Even for carrageenan, you know, for carrageenan we have only one company, a big company, and couple of small companies. There too we can simply produce less than 10% of our requirement. Imagine now all this requirement is has to be met through import. So how much our country is spending for import? Just now our honorable Prime Minister also has told us to be self-reliant, and seaweed also is one of the areas where our country has to be self-reliant. So all these studies basically are going to help us, and as I said, cultivation and processing, this is one of the technology developed by our institute, and I think I need not have to read everything, just to tell you that all this basically has formed a various products, you know, and you can see these products are not only for agriculture but even for fuel intermediates. So energy is one of the critical areas for the country, and seaweed can basically help this as well. I think this is my last slide. As I said, and we have identified opportunities, but there are constraints that, like, not many local seaweed species have shown robustness and adaptability, and we basically have to screen, and we have to have good species breeding programs, and all these constraints have to be overcome by this kind of research. Again, our biggest problem is those who are in South India and have seen commercial cultivation might be aware that after monsoon there is always a problem, because monsoon, you know, the sea is very rough. All your cultivation infrastructure had to be uh, brought out to the shore, and you need to conserve some of your biomass in the heavy wave action and all that. So you don't have good technology today for sufficient germplasm preservation, and I'm sure we are also working on it, and I'm sure this is one of the biggest constraint. Also, there is a low awareness. I don't know how many of you are phycologists or have phycology background. Those who have phycology background only know that what all are available, and you know, because they have studied this. And for other researchers, they may not be even knowing there are so many avenues. So there is a, this is the kind of situation for educated and, you know, people like highly educated people. So imagine, you know, what is the condition for a common man, a layman. So there is no awareness at all, and this is one of, again, big constraint. And as I said, there are a lot of opportunities. I don't think I should read again everything, but two opportunities I want to mention, that it has a big impact on carbon balance. And many of you might have heard carbon credit and carbon economy. So these are photosynthetic organisms, and they basically fix carbon dioxide, and commercial cultivation can give carbon credit to the companies which are in this field, and this is very big opportunity for the country. Also, those, as again I will say, that those who have seen commercial farming in Tamil Nadu, they will know that 90% of the workforce is women, and this is a biggest domain where woman empowerment can happen. So this is, I think, a big opportunity. And I think thank you for your patient listening. I feel that these biological resources become national asset when we develop a sound science and technology based applications. So with all this, I am thankful again, once again, to AARI management for inviting me to give this talk, and it is open for discussion if anybody has a question.
So I take this wonderful opportunity for thanking all the participants, and I take this pleasure of thanking Dr. Vaibhav A. Mantri sir for his excellent, wonderful insight presentation on the biodiversity, seaweed biodiversity in India. Thank you very much, sir. We would like to hear you more in upcoming seminars. Thank you so much for your wonderful time, and thank you for accepting our invitation, sir. Thank you, sir. Thank you for all the participants.
Yeah, thank you.
Yeah, I kindly request you to give your feedback. You will be if you are having any questions, please mail us. We'll try to connect with sir and get the answers. Thank you.
Yeah,
Thank you,
thank
sir.
you.
Thank you,
Thanks. We'll
sir.
close the session.
Yeah, we'll close the
Thank you
session.
so
Thank
much.
you.